Okay, just to briefly introduce myself to you guys, I'm very happy to be here and thankful to receive the invitation to give a talk today. So I am a Southern California native. I mostly grew up in a small town in Riverside County, Moreno Valley. From there, I went on to pursue my bachelor degree at California State University, Long Beach. I had an amazing time there. And I also had my first opportunity to participate in a research lab where I studied reproductive development in hamsters. I also had a fantastic opportunity to participate in a summer research experience here at UCSD while I was an undergrad. And subsequently, I applied to the program here. I'm also a recipient of the NSF Graduate Research Fellowship, which I am very proud of. So yeah, that's a brief introduction of myself. So in the Yelon lab, we are interested in understanding early patterning events during heart development and how those patterning events lead to cell type specificity in the different tissues of the heart. Many of the genetic mechanisms that lead to cell type specificity remain open questions in the field. So during this talk, I will address some of those open questions in regards to the specification of a group of highly specialized cardiomyocytes known as pacemaker cells. So pacemaker cells initiate the cardiac action potential. They are comprised of a small population of highly specialized cardiomyocytes at the venous pole of the heart. In mammals, this tissue is commonly referred to as the sinoatrial node, but in the zebrafish model, we also refer to it as the inflow tract. So the role of the pacemaker cells is to intrinsically depolarize and to set the, sorry, I'm just going to turn on my laser here, is to intrinsically depolarize and to set the rhythm of the heart. If endogenous pacemaker cells become dysfunctional, this can lead to arrhythmia, poor blood circulation, or even cardiac arrest. Current medical therapies rely on artificial pacemakers. However, this is an invasive and expensive remedy. So in order to relieve this alliance, there are many research studies that try to answer several open questions about how these cells develop. So the heart is one of the first organs to emerge during embryonic development. And in the zebrafish, the heart is actually comprised of 2 chambers instead of 4. So the Yelon lab utilizes the zebrafish as a model to study heart development for several reasons. So one is that the zebrafish heart is actually very visually accessible. In other words, we don't need to dissect out the heart in order to observe its development. The 2-chambered zebrafish heart is also an appropriate model to study pacemaker cell development because of the functional and molecular homology shared between the zebrafish inflow tract and the mammalian sinoatrial node. So this diagram illustrates the development of the zebrafish heart over time. And in this panel, we see the heart tube at 24 hours post-fertilization or 1 day post-fertilization. And as the heart grows and develops, the ventricular and atrial chambers begin to expand and loop. So when conductive activity is specifically disrupted at the inflow tract, or also denoted here as the sinoatrial region of the heart, this leads to, they found that contractile ability of both the heart chambers is affected. So this leads us to understand that the functional pacemaker cells reside in this tissue at the base of the heart, the inflow tract. So throughout this talk, when I'm referring to the development of the pacemaker cells, I will be mentioning the inflow tract. So the inflow tract, so the zebrafish inflow tract harbors these cells that are functionally comparable to the mammalian SAN. The zebrafish inflow tract and the mammalian sinoatrial node also share similar genes that are important for the function of this region. So here we actually see an example of a 4-chambered mouse heart, where the sinoatrial node is denoted by the expression of just one of the markers for the inflow tract, known as islet 1. And when we compare these images of a mouse heart to the images of a zebrafish heart, we see that that SHOX2 and islet 1 are also broadly expressed in the inflow tract of the zebrafish. In wild-type zebrafish, we see about 20 to 30 islet-1 positive inflow tract cells in wild-type zebrafish hearts. And also for this presentation, I will refer to the inflow tract cells as islet-1 positive inflow tract cells, as islet-1 is a genetic marker for this tissue. So considering the utility of this model, my project aims to understand how the zebrafish heart manages to establish such a functionally important population of cells at such a precise region of the heart. So what exactly makes these cells so special? Well, we know quite a bit about the downstream transcription factors that activate the pacemaker program, such as islet-1, which I've already mentioned, and SHOX2, as well as TBX2 and 3. Here, NKX2.5 is a gene that's important for the emergence of the atrial chamber. Thus, the inflow tract cells that are islet-1 positive are distinct from the NKX2.5 positive cells of the atrial chamber. So the upstream gene regulatory network is the transcription factor TBX18. TBX18 has been found to be necessary for the proper development of the inflow tract, but it's not sufficient on its own to specify the inflow tract's fate in cardiac progenitor cells. So the question becomes, what are the genes and signaling mechanisms that are required upstream of TBX18 to regulate inflow tract development? And secondly, when and where are cardiac progenitors specified to become inflow tract cells? So recent studies across the field have begun addressing these open questions. And one such study comes from one of our collaborating labs here at UCSD, the Chi Lab. So a postdoc at the Chi Lab used fate map analysis to characterize inflow tract patterning in the zebrafish anterior lateral, the ALPM. In this study, cells that were photoconverted at the lateral edge of the ALPM were appeared to emerge at the base of the atrium and were also positive for islet-1 and negative for NKX2.5. However, oppositely, cells that were tagged on the medial edge of the ALPM contributed to the outflow tract or at the top of the ventricle. So in total, the group saw that over 80% of the cells that were photoconverted at the lateral edge later contributed to the pacemaker fate at the inflow tract. So after fate mapping inflow tract progenitor cells to emerge from the lateral edge of the heart, the group then sought to identify the genetic factors that are responsible for activating inflow tract identity in this precise region. So the group used functional analyses to investigate the role of Wnt signaling pathway during pacemaker development. And here they used a PPT mutant, which is deficient in Wnt signaling. And they also utilized a heat shock promoter for Wnt5b to increase Wnt signaling. And they see opposite effects compared to the wild type. So when they decrease Wnt signaling, they see a decrease in the number of TCF positive cells at the outset of the region, and at 24 hours post-realization, they see a significant decrease in the number of islet-1 positive cells at the inflow tract. Oppositely, in the embryos with increased Wnt signaling, they see a higher number of TCF positive cells compared to the control, as well as a higher number of islet-1 positive cells compared to the control. So with these results, the group models a mechanism wherein the pacemaker inflow tract development or Wnt signaling activity at the periphery of the heart field promotes islet-1 expression through the simultaneous repression of NKX2.5. So what this data tells us is that Wnt signaling is very important for promoting the differentiation of pacemaker cells, but the question still remains as to which signaling mechanisms are upstream of Wnt, making sure that the right cardiac progenitors will become Wnt responsive in order to differentiate into inflow tract cells. So the Yelon lab has moved forward with several approaches to identify BMP signaling as a potential promoter of inflow tract development pathway. We've utilized the loss-to-Fin mutant, which harbors a mutation in a BMP signaling receptor to decrease BMP signaling efficiency. Here we can look at the expression pattern of several inflow tract inflow tract marker genes in wild type. And when we compare these images to the loss of fin mutants, we see a reduction in the concentration of the expression of these genes. Though the atrium appears much smaller in the loss of fin mutants, the loss of islet 1 cells is disproportionately smaller in these mutants. So comparing these two hearts imaged at 48 HPF, this data tells us that BMP signaling appears to positively regulate the number of inflow tract cells that are marked here in islet 1. But just how early BMP signaling is required in order to achieve just how early is BMP signaling required in order to achieve this proper development of the inflow tract? So that question can be answered by some previously generated data, also in the loss to fin mutants. So we see here in the wild type group that by 24 HPF, a population of 25 to 30 islet 1 positive cells in the inflow tract is established and is maintained through development. However, in the loss to fin mutants, we actually see beginning at 24 hours post-fertilization, we see a significant decrease in the number of islet 1 cells, and this decrease continues on as the heart develops. So what this data demonstrates is that BMP signaling appears to be required for the maintenance of inflow tract development. And also the BMP signaling appears to potentially have a role in progenitor specification prior to heart tube development at 24 HPF. So when we look at even earlier, even earlier stages of development, we also see a decrease in the number of islet 1 cells when BMP signaling is deficient. So in this figure, what we see is stages of DM addition, and DM is a pharmacological inhibitor of BMP signaling. And the earlier that BMP signaling is inhibited, the more drastic of a decrease in the islet 1 positive cells we see. So what the figure from the previous slide and what the figure from this slide show us is that BMP signaling appears to be required during these several discrete stages of development known as gastrulation to achieve proper inflow tract formation. What's also very important about this data is that in these several stages of development from dome to 24 HPF, in the field is known as the cardiac patterning stages. So to see that BMP signaling has such a drastic effect during cardiac patterning stages also suggests that this signaling pathway is important for the specification of progenitor cells. So I've also used an additional pharmacological inhibitor of BMP signaling known as DMH1. And using DMH1, which specifically targets the algate receptor, we also visualize a significant decrease in the number of islet 1 cells by 48 HPF. This data also demonstrates that there are potentially several mechanisms by which BMP signaling decreases or leads to proper inflow tract development. Additionally, we have used a morpholino against cordon. Cordon is an antagonist of the BMP signaling pathway. And using this morpholino to reduce the efficiency of the cordon function, we see a drastic increase in the number of islet 1 positive cells. So what this data demonstrates is that BMP signaling may have a dose-dependent role in properly formatting the inflow tract cell. So in summary, I've demonstrated that data that shows that Wnt signaling promotes the differentiation of the inflow tract, as well as the potential dual roles of BMP signaling to promote inflow tract cell development, a potentially early role in specification during gastrulation, as well as a later role in the maintenance of the inflow tract cells within the heart tube and through heart development. I've also demonstrated that levels of BMP signaling affect cardiomyocyte number and proportion as indicated in the loss to fin mutants. So this is our current model where we're looking at how BMP signaling may possibly play an upstream role to Wnt signaling in order for the proper promotion of inflow tract specification. Here is just an image of an embryo during gastrulation, where the distinct subgroups of progenitors may be affected by the BMP signaling gradients, where BMP signaling is highest, contributes to either the expansion or the decrease of the pacemaker progenitor pool. Okay, I'm just about at my time, so I just wanted to thank some of the members of my lab, as well as acknowledge some of my funding resources, as well as the PIBS training grant, formerly known as CMG. And if we have any, if the moderator says we have time, I'm happy to take questions, but I'll also stick around until the end of the talk for the Q&A. Thank you very much.